My name is Rajiv Mandal and I'm a scientific staff at Agropole. And our project is Benefits of Frass, which is a Frass project, which is a part of our Agropole Innovates larger project. In this project, we use the Frass, which is insect parts, and it's a manure from insects, which we utilize them and we analyze those frass, what kind of nutrients they contain, and afterwards we use those frass in those pot experiment to see what kind of result we are getting from those plants, how they are affecting those plants, and they are providing which kind of nutrients. Our project partners are the Agropole and the Brightland campus. Normally our pot experiment in the greenhouse, but we will do all the analysis in our lab. We started in the mid of February in 24. This project will end in August 2026, and there will be several other experiments we will do also in the field trial to see the benefits of Frass. That is our next experiment.
My name's Connor Watson. I'm a scientific staff member working here at the Rhine Valley University of Applied Sciences. I'm involved in the Frass Agropole Innovates sub-project as, an overseer of various Frass projects, designer of some experiments, whether in the lab or the greenhouse. Here the research question is whether plants, when they're fertilized with Frass, they suffer from over-fertilization or not. So here we've got, well, there were 3 different test plants. Radishes, they've been harvested, so we can't see them, but we've also got lettuce and peas, and they are receiving between 100% and 175% of the recommended nitrogen dose. An MSc student has set up this experiment with 7 replications of each treatment, so it's statistically very robust. The experiment was set up in February, and there seems to be a very plant-specific response. When you look at the literature, there's been lots of work done with maize, for example. We didn't want to repeat that. We want to look at some alternative plants, and there hasn't been much work about whether the plants get all of their nitrogen immediately or whether they can be provided with an excess without suffering. So that's really what we're looking at here. Is there a biomass penalty if you give plants more of this stuff with reference to nitrogen than you would perhaps with a conventional inorganic fertilizer? We plan to analyze, well, obviously the biomass production, the above ground biomass production of the plants. It's a bit more work, but we also plan to extract the roots from these pots so we can get an idea of the root to shoot ratio. We're going to look at the uptake of various nutrients, macro and micronutrients, into the above ground biomass. And time allowing, we'll also do some nutrient extraction tests on the substrate the plants are growing in, which is a substrate of low fertility, a mix of sand and soil. First results we have is just the biomass from the radish. So only preliminary results, but we're going to have detailed analyses from the lettuces and peas when they're harvested.
This is our ICP OES that we use to analyze the heavy metals and phosphorus present in our Frass samples.
Certainly this is what we call an auto-analyzer. A lot of the research we do with Frass focuses on nitrogen. This machine allows us to look at the 3 inorganic species of nitrogen as it goes into soil: ammonium, nitrate and nitrite. If we have a lot of nitrite, that is not a desirable quality, and we've been looking at whether we can stop this buildup of nitrite using nitrification inhibitors. If we have a lot of ammonium, that's generally no bad thing, but there's a potential for some of the ammonium to be lost as ammonia gas. So we want to get an idea of the relative proportions of the 3 using this machine, and we're also interested in looking at the main 3 organic forms of nitrogen in Frass, so chitin, urea and protein, and their relative proportions and how that affects nutrient release from Frass.